miniaturized version of ribosome found in Microsporidia. A research team led by MIMS Scilifilab research group leader Jonas Berandon, Umeå University, Sweden, uses cryo-electron microscopy to provide near-atomic details of the smallest known eukaryotic cytoplasmic protein synthesis machine, the Microsporidian ribosome. 150 years ago, the European silk industry was threatened by an unknown silkworm epidemic. At that time, Louis Pasteur was able to identify the source of infection and made important suggestions for treatment. Silk production in Europe survived. Today, a Microsporidian parasite is known as the cause of this epidemic, and silkworm diseases still cause more than $100 million United States dollars in losses to the Chinese silk industry every year. Microsporidiasis is not restricted to silkworms. The diverse phylum of the Microsporidia contains thousands of species with parasites for essentially every animal. At least 14 of them can infect humans. Particularly at risk are aquacultures, sericultures and honeybee populations, in which infections can wipe out entire hives, as well as immunocompromised patients. Microsporidia are a risk for the environment, agriculture and human health, and the U.S. National Institutes of Health, NIH, recently added the parasitic fungi to the list of emerging pathogens of high priority. Even if Microsporidia infections are among the most common parasitic diseases in all animals, relatively little is known about their fascinating molecular life, which is shaped by an accelerated evolutionary rate and extreme genome compaction. Together with researchers from the Rockefeller University and Connecticut Agricultural Experiment Station, Jonas Berandon, new group leader at the Laboratory for Molecular Infection Medicine Sweden, MIMS, has published a cryo-electron microscopy structure of the Microsporidian ribosome, which visualizes the effect of extreme genome compaction on an essential molecular machine, Nature Microbiology, 22 July 2019. Microsporidian parasites can survive as spores in soil, water and air, where they arrest in a dormant state. Once ingested by a host, they use a unique ultra-fast infection mechanism to inject the entire content of the spore into the host cell. Once inside of a cell, Microsporidia steal small molecules such as ATP from their host organism. This parasitic nature allowed them to shed many important genes, reducing their genomes to produce these small molecules. This compaction resulted in the smallest genome ever described in eukaryotes, even smaller than some bacterial genomes containing approximately 2,000 highly compacted genes. Microsporidia are the minimalists of unexpected findings. With the help of cryo-electron microscopy, the first step was to obtain sufficient cellular amounts to extract ribosomes. To overcome this challenge, Jonas Berandon teamed up with a Microsporidia specialist, Charles Vosbrink, from the Connecticut Agricultural Experiment Station, who grew the Microsporidium viromorphonecatrix in larvae of the corn earworm Helicoverpa Z, a pest that can cause major damage to cotton and corn crops. After extraction of spores from the host organism, ribosomes were isolated from them, and cryo-EM and mass spectrometry studies were performed in the laboratory of Sebastian Klinge, a ribosome specialist at Rockefeller University in New York, together with Mirjam Hunziker. This allowed the team to provide a near-atomic model of the smallest known eukaryotic cytoplasmic ribosome. In the studied organism viromorphonecatrix, the ribosomal RNA is approximately 30% shorter than the RNA in yeast and even 15% shorter than the bacterial RNA in E. coli. While it was known that the Microsporidian RNA is significantly smaller than the related yeast RNA, it was unclear if this compacted RNA still was able to bind all the eukaryotic ribosomal proteins. Surprisingly, despite the loss of some of their RNA binding sites, almost all ribosomal proteins were still present in the structure, some of them exclusively bound by other ribosomal proteins and not in contact with RNA anymore, explains Jonas. Compared to the closest related ribosome structure from fungi, Microsporidian ribosomes have lost only 2 ribosomal proteins. In one region of the ribosome, where several RNA elements have been removed, a previously unknown Microsporidia-specific protein compensates for the extensive loss of RNA and serves as a placeholder. During their evolution into organisms with highly compacted genomes, Microsporidia have removed essentially all eukaryotic expansion segments insertions present in the eukaryotic ribosomal RNA. The findings represent a reversion of the evolutionary expansion found in eukaryotic ribosomes. In eukaryotic ribosomes, expansion segments interact extensively with ribosomal proteins, and a loss of these elements could also coincide with loss of the proteins that are bound to them. The most surprising finding was that the characterized ribosome appeared to be functionally inactivated by 2 Microsporidian dormancy factors, MDF1, MDF2, and we can now assign a potential role to these 2 proteins of unknown function, says Jonas. Ribosomes were isolated from Microsporidian spores, the extracellular dormant spore stage of the organism. As the parasite depends heavily on the resources of its host, an efficient shutdown mechanism for cellular processes could be advantageous to preserve energy during the spore stage. One of these two identified factors exists in all